So the last 10 years, I've been involved in studying skeletal development, and up to now, up to the last 2 years, I was studying transcription factors, SOX5 and SOX6. But in the last 2 years, my research has taken a new direction, and I've gotten into the new and interesting field of membrane trafficking. And actually, I'm now studying its effect on skeletal development and disease. So in short, membrane trafficking is the process by which proteins from their site of synthesis, which is for most proteins the ER, get transported up and through the Golgi, where post-translational modifications occur, to their location, either inside the cells for proteins that are, for example, are involved in lysosomal function or endosomal function, or to the plasma membrane, or for plasma membrane proteins, of course, or outside of the cells for secreted proteins. The membrane trafficking pathway also includes the endocytotic pathway, by which proteins actually enter the cells. Now, membrane trafficking is a tightly controlled process to ensure that proteins end up where their function is needed. But no matter which trafficking event you look at, whether it's like from the ER to what would be the Golgi, or from the Golgi to the plasma membrane, Golgi to the endosomes, you can distinguish 4 steps. The first step is the budding of the transport vesicles, and it starts with a concentration of cargo at a distinct location of the donor membrane, which then subsequently starts bulging out under the influence of so-called cold proteins. And it leads up to the fact that the forming transport vesicle will separate from the donor compartment. The second step is the movement of this newly generated transport vesicle towards its destination. And it uses the cytoskeleton and motor proteins to aid its movement. The third part of a membrane trafficking process is the recognition of the transport vesicle by its acceptor compartment. This is the so-called tethering, and it's under the control of so-called tethering factors. These tethering factors will bring the membrane of the transport vesicle into close contact with the membrane of the acceptor compartment, and this will lead to the fusion of the membranes and the release of the cargo of the transport vesicle into the lumen of the acceptor compartment. And this process is controlled by so-called snare proteins, which are present both on the membrane of the transport vesicle and on the membrane of the acceptor compartment. Now, in the last decade, an increasing number of human inherited diseases have been shown to be caused by defects in this process of membrane trafficking. Now, a very large number of these are what can be called cargo mutants. They're actually caused by mutations in a certain protein that will lead to misfolding of this protein and to this retention in the endoplasmic reticulum, as the previous speaker has already mentioned. An example is cystic fibrosis, which is due to the retention of a CFTR protein. Another example is the skeletal dysplasia osteogenesis imperfecta, which is caused by misfolding of type 1 collagen and its retention in the endoplasmic reticulum. However, disease-causing mutations have also been identified in components that control each of the 4 steps of the membrane trafficking process itself. An example of a disease that affects vesicle budding is Anderson disease, which is caused by mutation in the cold protein SAR1B. And these patients, one of their phenotypes is that they have a defective generation of lipid transport vesicles. An example of a disease that affects vesicle movement is Charcot-Marie-Tooth 2A1, which is caused by mutations in the motor protein GIF1B. And one of their phenotypes is that synaptic vesicles in the axons of neurons fail to move along the cytoskeleton. Now, an example of a vesicle tethering disease is spondyloepiphyseal dysplasia tarda, which is caused by mutations in the tethering factor sertolin, which is part of a multi-protein tethering complex. So they have problems with actually getting the transport vesicles located at the destination membrane. And then finally, an example of a vesicle fusion syndrome is Setnick disease, which is caused by the mutation in the snare SNAP29. And these patients, one of their phenotypes is the accumulation of secretory granules, vesicles in the differentiating epithelial cells, because the fusion of these vesicles with the plasma membrane is not as efficient. Now, from studying diseases that are caused by mutations in components of the trafficking machinery, there is one surprising observation. The membrane trafficking process is essential for cell function. If you don't get your proteins to where they are needed, your cell will not be healthy, will not actually survive. But from the study of these diseases, we have found that defects in these components do not always lead to a global phenotype, but can be very tissue specific. Now, this, of course, is caused by the fact that there is a high level of redundancy in the membrane trafficking process, with often the expression of multiple isoforms of one given protein. So an example are the code proteins Sec23A and Sec23B. These are widely expressed, and they are involved in a generation of transport vesicles. Those Sec23A mutations only affect the craniofacial skeleton, causing craniolenticulosutural dysplasia. Apparently, in all other tissues, except for the tissues of the skeleton, Sec23B is able to compensate for loss of Sec23A. Now, the fact that this does not happen in the tissues of the skeleton, cartilage, and bone, is the fact that tissues which require high vesicle transport are more sensitive to disruption, of course, of the membrane trafficking machinery. Like cartilage and bone, they secrete an enormous amount of proteins to form the rigid extracellular matrix that is needed for their mechanical function. So this means a lot of proteins need to be transported through the cell towards the extracellular matrix, and this would require both isoforms, and when you only have Sec23B, it's just not enough, and you get a phenotype in these tissues. Now, another explanation why defects in widely expressed components of the trafficking machinery do not cause a global phenotype is that some of these proteins also have cargo-specific functions, that only tissues expressing this certain cargo will be affected. And the mouse model I'll be presenting the data from is a nice illustration of this third point. So the mouse that is shown in this slide was identified in an ongoing experiment in Dr. David Byers' lab, where ENU, so this is mutation, treated mice are screened for autosomal recessive mutations that affect organogenesis. So this mouse presents itself with a doomed skull, very short snout, and very short limbs. It also often has its intestines outside of the body. Now, most of these defects point to a severe skeletal dysplasia. And indeed, when we looked by skeletal preparations, we found that there was a severe delay in mineralization and linear growth of all bones. So in these skeletal preparations, the cartilage is stained blue with Alcium blue, and then mineralized cartilage and mineralized bone stains red with Allicin red. And you can see that at E17.5, there is no mineralization in the mutant mice. But 2 days later, when the mice are born at P0, you can see mineralization is present, but clearly this element is much shorter than the wild type. So we looked in more detail by performing histology, and you immediately noticed that when you look at the mutant, that the cells throughout, so this is a cartilage, this is actually a developing humerus, all these cells are swollen, have increased in size compared to what you see in the wild type. Now, the reason for this cell swelling became obvious when we did electron microscopy. We noticed that the mutant chondrocytes, so the cartilage-producing cells, all have a swelling of the ER compared to what you see in the wild type. In this ER swelling, we also saw in the bone-forming cells, the osteoblast, you see an increase in ER volume compared to what you can see in the wild type. Now, interestingly, this ER swelling was only visible in these two tissues. Any other tissue that we looked at, for example, endothelial cells, mesenchymal fibroblasts, or myocytes, they do not have this swelling of the ER. Now, the consequence of ER swelling is that you get the triggering of what is called the ER stress response. The cell tries to deal with this retention of proteins in the endoplasmic reticulum. And you can monitor this by checking the expression of BIP, which is an ER stress early response gene. And you can see that in the mutant, so this is again a 15.5 humerus, you see that it's a much stronger expression of this gene in the cartilages, the same for the ear. Now, a consequence of ER stress is that it targets cells for apoptosis. And not surprisingly, we found that there is massive cell death in our mutant cartilages. So this, of course, explains the severe skeletal dysplasia with the massive death of cells in the developing bones. Now, we had this swollen ER cisternae. Now, the retention, so this is most likely due to the fact that proteins are getting stuck in the ER and not being able to be transported out. Now, it's what you often see when you have a cargo mutant of a cartilage or a bone-specific protein, for example, osteogenesis imperfecta, type 1 collagen mutations that result in the retention of collagen in the ER. The osteoblasts of these patients actually have a swollen ER. The same for metaphyseal chondrodysplasia type Smits, which is a mutation in collagen 10, which leads to misfolding of this protein. These patients, the chondrocytes, have also a swollen ER. So when we looked at this, we thought that the ENU mutagen had induced a mutation in a cartilage or bone-specific protein, which prevented secretion. Now, using SNP panels, we were able to narrow down the candidate region for the ENU-induced mutation to a 4 megabase region on chromosome 12, which contained several genes, which are shown here. But none of these genes are either cartilage or bone-specific. None of them had a known role, at least in the published role, in skeletal development. So we basically sequenced all the exons of all these genes just to find the ENU mutation. And we ended up identifying a single nucleotide change in the gene TRPL11, which stands for thyroid receptor interacting protein 11. And using an antibody that recognizes an epitope and terminal of the mutation, we found that we actually have no protein in our mutant mice, and that this ENU mutagen had created an effective null allele. Now, TRPL11, despite its name, it's a membrane trafficking protein. It belongs to the Golgi protein family, and it localizes on the cis side of the Golgi, where cargo vesicles from the ER are accepted. It has been postulated to be a tethering factor. So it would dimerize. So this is actually one other name of TRPL11 is GMAP210. So the protein would dimerize, and with its C-terminal grab domain, with the aid of a few other proteins such as RF1, it would actually bind to the membrane of the Golgi. And then the central region, which contains lots of coated core, would form a rod-like structure, which it would protrude into the cytoplasm. And then with its N-terminal region, which contains an amphipathic lipid packing sensor, it would be able to recognize curved vesicles. So this would mean transport vesicles that are coming from, in this case, from the ER. So it would then bind these vesicles and then help in the fusion of these vesicles with the plasma membrane of the Golgi. Now, the Spanish group, Rios and co-workers, so about 5 years ago, they did siRNA knockdown of TRPL11, and they found that TRPL11 is actually needed for the maintenance of the Golgi structure. So they saw that when they added siRNA, all the Golgi marker proteins started to dispersing, to spread out in the cytoplasm. So we had a look by EM at the Golgi structure in our affected, in our mutant in affected cells, and we indeed found that in the chondrocytes, there's a complete absence of the Golgi structure. So you normally have this nice stack of flattened cisternae, and you cannot see this in a mutant. You have a collection of vesicles. Now, TRPL11 is widely expressed. It's not a single cell type that we have found that does not actually have TRPL11. So this is an illustration of it. So this is an in situ hybridization using a TRPL11 specific probe. This is a 15.5 humanist. You can see the strong expression in the cartilage, strong expression in the bone, for example, also in this ligament and also in the muscle. So when we started looking at the Golgi structure in other tissues, because based on the in vitro studies, it was postulated that this protein would be really essential for maintaining the Golgi structure. We noticed that some tissues have defects, but not as strong as what we see in the chondrocytes. For example, the Golgi cisternae in the kidney are swollen, but they still have their more or less a stacked organization. However, other tissues, which also express TRPL11, have an absolutely normal Golgi structure, so no disruption of the the stacking. So we have an ubiquitous expressed protein, which in vitro was shown to be necessary for forming of the Golgi stack structure. But when you knock it out in vivo, when you're doing in vivo study, you'll find that it is actually very tissue specific, which illustrates the importance of doing in vivo studies for understanding, getting a full understanding of what the protein is actually doing. Now, one of the cell types in which we did see phenotype is the primary skin fibroblast. So this is a confocal picture of the Golgi stained with a Cis-Golgi marker, the G130, and a Trans-Golgi marker, actually RL1. And this is how a Golgi looks like under a confocal. So it's not actually like what you could see, the nice stacking. It's a reticular network. But when you look in the mutant, you can see that this network is completely gone. The Golgi is basically collapsed around the nucleus, so this would be the nucleus. But what we can see here is that despite this loss of structure, the organization of the Golgi is still maintained. There is no overlap between the Cis-Golgi marker or the Trans-Golgi markers. You see, there is no yellow color here. Now, because of this disruption of the Golgi structure, in the tissues in which we could see the ER swelling, we thought, okay, Golgi structure is disrupted. The efficiency of transport of proteins through the Golgi will be less efficient, or will be blocked, and this leads consequently to a backup of proteins in the ER, explaining our phenotype. So we wanted to see whether this was indeed the case. So we did a VSVG GFP transport assay. So this protein is a temperature-sensitive mutant, which when cells are grown at a non-permissive temperature, like 40 degrees, this protein gets stuck in the ER and will not move to the Golgi. This is what you could see in the infected cells of both the wild type and the mutant. And you can see that there is the Golgi marker G130. There's no overlap with the green signal of the green fluorescent labeled VSVG. But when you put these cells at a permissive temperature of 32 degrees, the VSVG is released from the ER and will actually move to the Golgi. And interestingly, this happened both in the wild type and the mutant. The pattern of VSVG is now identical to that of the Golgi marker. So there is no blockage, or at least no general blockage, in the transport of proteins from the ER to the Golgi. So we were asking, what is causing this ER swelling? Is it just the amount of proteins that need to be secreted by these cells? Or is there an impairment in the transport of a specific protein or specific proteins? So we started looking at deposition of the extracellular matrix proteins in cartilage of our wild type and our mutant, and by immunofluorescence. So when we looked at the type 2 collagen, there is actually no difference. Couldn't see any difference in deposition. Type 2 collagen seems to be able to exit the ER and be transported to the extracellular matrix without any problems in the mutant. And the same thing is true for aggregate. Now, these two proteins are the largest and the most abundant ones produced by cartilage. So we thought these were the most likely candidates of getting stuck in the ER. However, it's a much smaller protein, perlecan, that gets accumulated. So this is also a proteoglycan. So this is a wild type and a mutant again, and you can see that the staining is different in the mutant. It's much more localized, much more intense. And when you look at the confocal microscopy at the wild type and mutant cells, you can see that the red signal, which is the perlecan, will, in the merged picture at least, will overlap with the green signal of cal reticulin, which is an ER lumen marker. So perlecan gets stuck into the ER. Now, perlecan is produced at high levels by chondrocytes, but it's also produced by osteoblasts, and it's a prominent component of the basement membrane of the epidermis. And when we looked at the epidermis, where perlecan is produced by dermal fibroblasts, we found this dotted pattern indicating like accumulation of perlecan inside the cells of dermal fibroblasts. So this fits very well with with our phenotype. Like we have perlecan, which is expressed by chondrocytes, cartilage, and dermal fibroblasts. And in each of these three cells, we see a phenotype. So in summary, the absence of 1111 does not impair secretion of the most abundant or the biggest proteins that needs to be secreted. However, it seems to affect the secretion of specific proteins. And as I just mentioned, this could explain at least part of it, of the tissue-specific phenotype when you inactivate this protein. Now, I've been talking here about our mouse model, and you can wonder what this has to do with rare diseases. So when we compared our mouse phenotype with the phenotype of human syndromes of which the genetic cause was not yet identified, our attention was drawn to achondrogenesis type 1A. This is an autosomal recessive neonatal lethal, short-limbed dwarfism, and is characterized by an undermineralization of the skull and the vertebral column seen in this X-ray. So this fits very well with what we observed in our mouse model. This is an example of the undermineralization of the vertebral column in our 1111 mutant mice. So we dug up some old electron microscopy pictures of an achondrogenesis type 1A patient, and we noticed that the chondrocytes have also this increase in ER volume that we see in our mutant. So we obtained DNA from 10 different patients and started sequencing the 1111 gene, all the exons. And in all 10 of them, we found that there were gene inactivating mutations, indicating that 1111 is indeed, mutations in 1111 are indeed the underlying cause of achondrogenesis type 1A. So in final summary, our research has shown that absence of 1111 leads to the accumulation of cargo into the ER, leading to ER stress and subsequent apoptosis of the cells of the skeleton, which induces the severe skeletal dysplasia. We've shown that absence of 1111 differentially affects the secretion of specific extracellular matrix proteins, so that there is definitely cargo specificity. And an important point is that 1111, despite being ubiquitously expressed, is required for the formation of the Golgi stacks in some but not all tissues. And this illustrates, again, the importance of, as I mentioned before, of doing in vivo studies. Because based on in vitro studies, I would have thought that this gene would have disrupted the Golgi structure in all cells. And then finally, by comparison of our mouse phenotype with that of human diseases, we found that 1111 mutations cause human achondrogenesis type 1A. And this is a nice illustration of the usefulness of forward genetic screens using model organisms for the identification and study of human diseases. So with this, I would like to conclude and acknowledge all the people that were involved in the study. As you can see, this was a highly collaborative study involving several research groups. So I'll be happy to answer any questions.
Feel free to use the microphones, and there is a switch at the bottom of the microphone that you can turn on, and it lights up red, and you can ask your question.
Patrick, very nice. Do you have any kind of sense of how you do cargo selection in these sorts of things, in these situations?
So that's still one of the questions, of course, we need to figure out is how this protein actually recognizes a specific cargo. So it might be that, like when you generate cargo vesicles, like some cargo proteins are actually inserted into the membrane of the vesicle. And it might be that 1111, either directly or through interaction with other proteins, then would be able to bind or efficiently or more efficiently these vesicles with this specific transmembrane cargo. But Perlecan, for example, it's not a transmembrane protein, so it will be in the lumen of the vesicles. But in these vesicles, there's always complex formation, like one cargo protein will interact with another cargo protein. So that is one way that it would be possible to actually get like a very specific cargo selection. But the truth is, we actually don't really know how actually 1111 is doing this cargo specificity.
Questions? Please.
Perlecan is the only cargo for 1111. What happens to the other 1111 cargos in other tissues?
So we're still looking for other cargos. Well, the problem is we wanted to do these studies on like on chondrocytes, because they have this massive swelling in the ER, and then we were thinking of just getting the ER out, running it to a mass spec, and see all the different proteins that get retained. But we cannot culture these chondrocytes. It's like it's very frustrating. It's like so the mouse usually they die at 17.5, which makes their ribs cartilages too small to actually get enough chondrocytes out of there to do primary cultures. And even if the few chondrocytes that we get out of there, they fail to grow. So we can't get enough material to actually do this mouse spec, which is why the VSVG transport assay was done on primary fibroblasts. So we've always, so far, been trying to identify them using antibodies, but good antibodies are not always available for all proteins. So we're now making the conditional knockout, and then we are thinking of just getting the cartilage out of these mice, getting these alleles, and then inactivate it in vitro by adding adenovirus 3 and see whether we can mimic the phenotype and then go after the cargo. But I would expect that it's not just perlecan, but that a few other proteins would actually get stuck in ER too.
Perlecan is exclusively
see, I think it's for like, yeah, that comes back to the same point. It's probably not only perlecan. It's almost maybe some other protein that we haven't found yet, but it also gets stuck. But it's like to induce ER stress, you need this massive increase in volume. Now, perlecan is very strongly expressed by cartilage, so it might be that just by itself, it's enough that it gets retained, that you get this swelling in the ER. But as I said, it's most likely that a few other proteins get stuck, and that's why you get this ER swelling. To a critical point where there is ER stress response. I haven't done it yet. I don't know whether there's a good antibody against it or not.
I'm just wondering if it's
yeah. Well, it doesn't seem to inhibit the transport of aggrecan.
well, it's chondrocytes.
And that's a chondrocyte, yeah. So, and then perlecan, yeah. Yeah, but I have to look at them. I haven't. If there is a good antibody for it, I can easily look at it.
It's a good antibody against it.
Synica. Is it commercial?
Great. Thank you, Patrick. Thank you very much.